third cell cycle of mitosis. It is a period of time that starts when the cell is formed until it divides. It includes two phases, interphase about 19 hours and mitosis about 1 hour. But this time of duration for interphase and mitosis is variable. Let's look at this graph that shows the variation in the mass of a chromosomic material per cell in arbitrary unit as function of time during cell cycle. Each cell cycle consists of interphase followed by mitosis, or each mitosis preceded by interphase. Okay, now during interphase, the mass of a chromosome material increases from one arbitrary unit to two arbitrary units. Okay, then we conclude that the chromosomic material duplicated during interphase, which we have talked about it at the beginning of the lesson. Then, during mitosis, that consists of four phases: prophase, metaphase, anaphase, and telophase. The mass of a chromosomic material decreases into half from two arbitrary units into one arbitrary unit. Now, in details, evolution of a chromosomic material. First, interphase consists of three phases: G1 or first growth phase, S, which is called synthesis phase, G2, which is second growth phase, and mitotic phase, which represent for mitosis, which is prophase, metaphase, anaphase, and telophase. During G1 phase, which is the beginning of interphase, the chromosome consists of one chromatid. Then it's followed by S phase, which represents the synthesis phase, which the DNA is start to replication and appearance of replication bubbles. Then the end of interphase is called second growth phase, which represent the chromosome consists of two sister chromatids, or this chromatid is like filament. Okay, then from G2 to prophase, what is the major event that takes place? The chromosomes are condensed and still consist of two chromatids. Okay, then prophase followed by metaphase. The chromosome occupy the equatorial plate. Then anaphase, where the sister chromatids are separated and migrate to opposite pole, which is called polar ascension. Then at the end of mitosis, which is telophase, we obtain two daughter cells. Each one contains two chromosomes of one chromatid. Okay, between telophase and another cell cycle, or to start with another interphase, the chromosomes are decondensed. Okay, this document represents the evolution of the quantity of chromosomal material during one cell cycle and the reproduction microphotography of cells during mitosis. As shown in this graph, during interphase, the quantity of chromosomal material duplicate, where at the beginning of interphase G1, the chromosome consists of one chromatid, while at the end of interphase, the chromosome consists of two chromatids. And the difference between G2 and P, which represents the prophase, the chromosome is condensed. Then at the end of the mitosis, at telophase, we obtain two daughter cells that contains that contains two chromosomes which identical to the initial cell at G1. Conservation of the genetic information by undergoing successive mitotic divisions, the zygote form all the cell of an organism. For example, at day 1, okay, the zygote consists of one cell. Okay, after undergoing first mitotic division, it becomes two cells. Then second mitotic division, we obtained 4 cells. Then 8 cells, 16 cells, 32 cells, etc. In order, for example, at day 4 or 5, the zygote become morolla, then blastocyst. Okay, in this case, during these divisions, there is reproduction of identical copies of a chromosome. In another word, we obtain same karyotype for all body somatic cell in all days. Okay, يعني in another word, all body cells have same karyotype. What we can conclude? We can conclude during mitosis from zygote to morolla to blastocyst to individual. During mitosis, the number of chromosomes and the genetic information are what are conserved. It's important word. The genetic information and the number of chromosomes are conserved. From interphase to cytokinesis, the entire process of cell division can take on average 10 to 20 hours in a typical plant or animal cell. Depending on the nature and use of the cell, the process can happen at different frequencies as well. In humans, our skin cells have a high turnover rate due to wear and tear and go through mitosis very frequently, whereas other cells such as adult neurons and muscle cells rarely divide. This process allows for growth and repair to prolong overall physiological as well as life itself.